Ladies and gentlemen, moving on to our next session. Our next topic is bringing sci-fi to reality with CRISPR genome editing by our expert in the business. May I now invite Dr. Park Poom Sapsunthorn, researcher at MIT and Futuristic Research Cluster of Thailand. And may I invite Kun Nika Chomwong, ex-CEO, Sugar Logics, to join us on the stage as a moderator. A round of applause to welcome both of them, please.
Hello, everyone. My name is Kalika, and I just want you guys to close your eyes for a second. So, imagine a world where Alzheimer's is completely eradicated and children with genetic disease who destined to die before the age of 5 can basically be cured entirely before they're born. And, some beneficial traits like strength and cognitive ability can be programmed and passed on to the next generation to no longer rely on nature and have full control of how we choose to have our species evolve. I'm talking about rewriting our own biological destiny, and whether you like it or not, it's no longer a science fiction, and the possibilities of all I mentioned and more is already knocking on our door. So today, Dr. Pakum will be introducing CRISPR, of course, which is one, as an entrepreneur and biologist myself, I would argue it's one of the greatest breakthrough of the past few decades. So please welcome.
Hello. Thank you, Kun Kulika. So I think CRISPR has been a buzzword in biotech for the past 4 or 5 years, and it started to make a news to the general public probably like the last 3 or 4 years. Some of you probably have heard of it, but I'm not sure about like what is the actual potentials of this technology. So I will spend about like 10 to 15 minutes today talking about the potential of these technologies, and then we'll have a quick fire chat discussion about some of the related questions on these technologies. So, sorry. Let's wait for the slide. Okay. So when people think about the potential of CRISPRs, we have to go back to the basic of biology first. All living things, all life on this planet have genetic materials, which we call DNA. Right? It's the same type of materials that we have. Bacteria has, most of virus, or plants, microbes, fungus. All these species have the same language of life. Now, okay. Okay. Maybe I just talk about the thing for now. So okay. While we're waiting for the slide, I'll just talk about like the general stuff first. So basically, like for the technologies, because all the genetic materials that's on the planet Earth, most of it, except for a few types of virus, are made from DNA, and so any technologies that would allow us to go in and change the information on the DNA the way that we like, the way that we wish, we have the impact across the board. We have the impact on any industry, any types of applications that is related to living things. You can talk about medical applications, because of course our cells in our bodies also have DNA as a genetic materials. You can talk about agriculture, because our life in the farms from crops and farm animals and plants have DNA as genetic materials. Now these technologies is about 4 or 5 years old. Before that, we have other technologies that allow us to change genetic material inside the cells, but it's much more difficult to do. It takes much longer times. It requires a lot of money in order to go into a specific locations on the DNA before we can change it the way that we want. So for that reason, CRISPR become the technology that is very powerful. Now, if you go back to the basic of biology of CRISPR, basically, it's originally an immune system of a bacteria. On the surface of the Earth, over the past, I would say, like 3 to 4 billion years, the planet is occupied by bacterial species, like billions of them, and over the course of the life of these bacteria, they have been infected over and over again by their enemy, which is a virus. Sometimes we call bacteriophage. And bacteria like us need to have some type of mechanisms that allow them to protect themselves from those viruses. Okay. So we already talked about this. I give the intro about DNA as genetic materials, but I would like to put this in a perspective a little bit. We learned that from our middle school or high school about DNA as genetic materials, but about editing the DNA at a specific locations is actually more difficult than a lot of people imagine. Let's imagine this. Most of the time, the size of the DNA, the size of the sequence location of DNA that we would like to edit is pretty small. Sometimes when things go wrong on your genome, on your genes, it's about 10 to 15 bases. And the size of our genome is 3 billion bases. So if we put that into a perspective, imagine a long road from the southernmost part of Thailand to the northernmost part of Thailand. Okay, imagine that long road, and imagine that you drop a coin the size of a 1 baht coin somewhere on that road, okay? And you want to find that coin at that location on times, okay? That's the level of precision that usually need when you're talking about changing the genome for many practical purpose. Now, when you talk about CRISPR, as we know, DNA is genetic materials, and for a lot of bacteria on the planet, it have some immune system that allows it to prevent foreign DNA that somehow get into the cells, DNA from virus or some other bacteria that invade into the cells. And the way that bacteria do it, for some bacteria, it has a mechanism that we call CRISPR. And the way it works is that at the sequence locations of the virus or the foreign DNA, the bacteria can produce 2 types of molecules. One is the RNA, another one is the protein. And these 2 molecules can come together at that specific locations, and it will cut that piece of DNA. Okay? And the way it works in natural system, when the DNA is cut, the virus or that foreign DNA is destroyed. Now, we humans look at this phenomenon in nature, and we adapt this type of phenomenon into our technologies that allow us to cut the DNA at a specific location. Okay? The reason why this is powerful is that in the past, when people want to cut DNA at a specific location, they need to make a new enzyme. They need to make a new protein. But it's very difficult to make a new protein or enzyme because the protein has a very complicated shape, 3-dimensional shapes, and it usually takes months or years to engineer a protein that function properly. The reason why CRISPR is powerful is that you use the same enzyme. You can use the same enzyme over and over. The only part that you need to change is the second molecule, which you call the guide RNA. the size is much smaller, the structure is much simpler, okay? And usually you can synthesize it and test it in a matter of days, okay? And the cost is like 1,000 times cheaper than the previous generation of DNA targeting technologies. So that's why CRISPR become very popular over the past year. Now, you may ask, okay, once we cut the DNA, what's going to happen next, right? There are two possible ways that things can happen. When you cut the DNA in the cells, especially in the higher organisms, like humans or animals or plants, they will try to fix that broken DNA. And they will try to, when they fix that DNA, one way of doing it, they just rejoin that piece of DNA together. But most of the time, this process is not perfect. So you're going to have some error at the junction, okay? So for this reason, if you use this to cut at the genes or any position on the genome that you don't want, during the process of repairing DNA, you eliminate the function of that gene. Okay. That's one application of the technologies. Another application is that if you do this, if you cut it and you also supply in the cell with another short pieces of DNA that is similar to the DNA at the junction that you cut, that pieces of the DNA can go in and basically replace the sequence of DNA at the broken junction. So this way, you can edit or change the sequence of the DNA at the location. Okay. Now, that things happen in like 2012, 2013, that people discovered these mechanisms. And after that, people come up with many other technologies that build upon this one. For example, there's many research groups that figure out if they can disable the cutting function of the enzyme so that the enzyme can still bind to the location on the genome that you want to bind, but it's no longer cut it. And then you can augment these enzyme molecules with other functional modules. For example, something that can allow you to activate the genes that you bind or inhibit the genes that you bind, or some functional modules that allow that location to have a light or to have some kind of protein that give out a signal. Okay, so this way you can also use this technology to turn on or turn off any genes that you want at that location that you choose, and you can also use it to look at the specific location of your genes in the genome. Now, this slide is really complex, and I'm not going to talk about a lot of detail, but just to show you that there are actually two branches of technology that came together when CRISPR bonds. One is a technology for genome editing, which has been around since I would say from this slide it's like 1970s, but some people say it went back far beyond that. It has been around for a long time, but as I said, it's not really efficient in the past. And then there's another technology, which is a CRISPR, which people study the basic science of it for a long time. But only in 2012 that the two things come together, and people figure out that now we can use this immune system from bacteria as a cutting tool to target the genome. Now, this is just a slide to see, the buzz and the excitement that has been going on in the field over the past 5 years. These technologies come out. These are just tiny fractions of a startup company that has emerged over the past years. These are the rise of the funding in the U.S. from the NIH on the CRISPR-related research, and also the number of projects that emerged over the past years. And also here is the number of publications that skyrocket over the past years when the technology takes off. So basically the excitement has been going on for the past few years, both in the academic sections and also in the industrial world. Now let me show you several exciting applications that has come about over the past several years. The most obvious one is gene therapies. If you can edit the DNA at any location that you want, the first thing that you might want to do is you want to go to sick people that have problems at the genetics levels and then fix the base or the genes that cause the problem. For example, this is some of the news that came out just half a year ago. There was a group from Harvard that started to use this tool to cure deafness in mice. It turned out that about half of the deafness in mammals is actually genetic related. And there's a genes that when it's expressed, it will destroy the hair cell in your ears. And this research group figure out, if you can disable that genes and prevent or prolong the onset of this deafness in mice. There's many other cases that related to gene therapies. People use it for treating the vision loss. People use it, starting to use it in curing ALS, cystic fibrosis, sickle cell. This one is a disease related to your red blood cell. There are many other examples of this, and they come down to the same theme, which is there's something wrong in your genomes, and you use this tool to change that gene that has problem into the correct copy of the genes. Now, none of these products are in the market yet, but there's a few cases that are already on clinical trial. For example, I think that the one that's going to be on clinical trial this year is the one related to thalassemia. There's going to be one for sure in Europe. In the US, just have a news a few days ago that a few weeks ago that they're going to postpone it. But if you go to place like China, they probably have been trying this for a couple of years already. There are some other applications that related to genetic disease, like HIV. So if you know HIV is a virus that once it infects your body, it will destroy your immune system, right? And over the past several decades, people came up with technology that can suppress the virus or reduce the reproduction of the virus. But the problem is that this virus can hide its DNA piece on the genomes of humans. And once it gets into the genome of humans, it's really difficult to get it out. So it's really difficult to kill HIV completely, right? And one of the applications of CRISPR that people have been working on is that they use this enzyme to go into that location that virus hide itself in the genome and just cut out the virus from the genomes. There's a case that people use it for farm animals. There was a pretty interesting paper from last year. One paper is about making pig less fat. Okay. So the idea is that the reason why pig is fat is that about 100 million years ago there was a mutation that happened in the ancestor of pig that prevent it from metabolize fat efficiently. Okay, and the consequence of that not only make pig fat. Okay, but another consequence of that is that pig cannot produce heat efficiently compared to many animals. So in pig farm, there's a lot of piglet that dies, especially in the northern countries, because of the coldness in the farms. So you can fix that genes, allow pig to burn fat more efficiently and produce heat more efficiently, then you can solve both problems at the same time. And so there was a research group, I think it's in China, that tried to fix that genes and allow pig to be leaner and also more tolerant to coldness, to low temperatures. There's another case in which people try to use this to cure pig from a hidden virus. There's some type of virus that hide in the genomes of pigs since many million years ago. And when people want to use pig for organ transplant, because in many applications in medicines, we use pig organs as a xenotransplantation to use pig organ in human, because we and pig have a pretty similar physiological system. Okay, but the problem with that is that there's this hidden virus in the genome of pigs. So the way people fix it is that they also use CRISPR to go into the genome of pigs and try to eliminate as many virus as possible from the genome of pigs, so that once you transplant the pig organ to human, you don't have to worry about this infection from the virus that's hidden in the cell. And of course, if you can edit humans, so why don't we go in edit babies, right? This is going to be one of the most controversial topics that will be discussed in the next sessions. If we can cure humans from any genetic disease, if we can make anyone tall and handsome and smart, should we do it? Should we not do it, right? People have been discussing this. I think they have already been trying that in China, in the US and European countries. They're still debating whether or not they do it. But that's also one of the things that is already in the realms of possibilities within the next, like, we'll say 5-10 years. This is some clinical trials that has been going on. As you can see, the black one here is in the Europeans and US. Right? The red one is in China. So the good thing about China is that China doesn't care much about ethics or regulation, right? So they just went ahead in terms of technologies and clinical trials compared to many other parts of the world. If you can edit human or animals, you can also do it with plants, right? This one's the news. Just a few weeks ago in China, they used it to increase productivity of rice, right, by editing the genomes. This one is a company from the UK that working on bananas. As you know, when you grow bananas anywhere in the world, the genetics of banana is pretty much the same because you grow it from the seed, right? You grow it from the shoot, right? You clone it from one banana to another. Because of that, banana is really sensitive to a lot of infections, and people try to use like CRISPR technologies to increase the diversity of the genes in banana to make it more tolerant to infections. And also coffee, we talk about decaf coffees, right? Is there any way that people can go in and change some pathway, eliminate some mechanisms in coffees, in the coffee plant, so that it can no longer produce or reduce the expression of caffeines, right? But but can maintain its aroma and its flavor. One of the most interesting thing about CRISPR application in agriculture is that it's going to change the landscape of the discussion about GMO. For many countries, including the US, they classify CRISPR-edited plants in the different categories from GMO plants. Okay, the reason for that is that when you talk about GMO plants over the past decades, you're talking about cutting and pasting the genes from different plants or from different type of cell into your crop of interest, right? But when you talk about CRISPR, you're not cutting and pasting any foreign DNA into the cells. You can do it just by changing a few bases here and there. For this example, it just passed the FDA in the US earlier last year. This one is a mushroom. When you buy mushroom from the market and you leave it in the fridge, right, it will turn brown. Right? That happened because there's some enzyme in the mushroom that caused that. So there's a company in the US that used CRISPR to change only two bases, only two nucleotides among millions or billions of nucleotides in the mushroom. Change only two bases to reduce the expression of that enzymes and make the mushroom remain white for a longer period of time, right? And the US FDA said, like, okay, that's fine. They're not going to regulate it, right? Because it doesn't require any transplantation of a piece of DNA from different organisms. Now, people are also getting more and more creative about how you can use CRISPR. So when you talk about CRISPR, the first application that people thought about is genome editing, right? But because CRISPR, if you look at it from another perspective, is a system that you can go in and bind to a specific sequence of DNA or RNA anywhere that you want. So there's a group of researchers that figure out a way that you can use it as a tool for detecting piece of DNA or RNA and classify them based on the types of disease. okay? Because for different types of virus or bacteria, they have different genetic materials. And if you can go in and cut at the locations and somehow use the consequence of that cut to report back to the researcher or to technicians. For example, when you in this case, in this news, basically they have a CRISPR that once it targets the location in the DNA, it will trigger another enzyme that change the color of the solution. Okay, so you use it as a tool for detecting disease, and this is a mock-up prototype of this company. It's called Mammoth Bioscience. It just found like earlier this year. So basically, you have these strips of paper that have the mixture of CRISPRs that target particular types of DNA or RNA from the virus or bacteria. And once it detects and cut that piece of DNA, it will create a different patterns of color in the piece of paper, and you can scan that with your mobile app, for example, and you can use it as a way to diagnose, the type of disease. Another angle of medical application for this is for antibiotic resistance. Most of the time, people talk about CRISPR, you're talking about cutting and fixing the genomes, right? But for many type of cells, especially for bacteria, when you cut the genome, the cell will die because the repairing mechanism is not so good. So the people, there's a lot of people in the field of antibiotic resistance. They are working on repurposing CRISPR into a tools that allow you to target a specific type of bacteria that cause disease without killing other bacteria that could be very similar to that, maybe like 1 or 2% difference in terms of genomic sequence. So why this is important? Because most of the time when people use antibiotics kill many types of bacteria, and it has a lot of side effects in terms of microbial ecologies in our bodies or in the soil, and it can enhance the process of antibiotic resistance. But if you can target a specific group, a specific strain of bacteria that cause disease without harming other bacteria, it can be a game changer in terms of antibiotics. This one is another pretty crazy application of CRISPR. This one came out in the mid of last year. So people use CRISPR as a way to store movies in the living cells. This one is a little bit difficult to explain, but the way it works is that the natural version of CRISPR, the way it works, it takes a piece of virus and it stores on a different location of in the bacterial genomes, so that in the future you can use this material from virus as a way to target the virus that infect the cells in the future. So basically, there's some mechanism in CRISPR system that allow you to store a short pieces of DNA into the genomes of the cells. And this research group, I believe it's from Harvard, so they encode informations, can be movies, it can be GIF image, it can be a song, it can be anything, into a sequence of DNA, right? You can think of sequence of DNA as what is called when you talk about digital information, it's a binary bit, right? It's 0 or 1. In DNA, it's ATGC. So basically, you just convert the binary bits into ATGC, and you can store that as a piece of DNA in the genome of bacteria. You can grow bacteria for many, many generations, and then when you sequence back, you can convert the information back to a movies, to a song, to whatever. Right? The reason why this is important is that you know that there's an explosion of data in the world, and in some application, you want to store data for a very, very long time. Think about your hard drive. It won't last longer than, like, I would say 10 or 15 years. Right? But if you store it on the DNA, it can last for like tens or 20,000 of years. In a good condition, if you can propagate it also in the living cell, it can last for millions of years. Okay, and so that could be another way of storing information for a very, very long time. There's also another group of people who are working on engineering the ecologies. This one is a technology that we call gene drive. Earlier, the past centuries, people discovered that there's some copy of genes that can prevent certain species of can prevent a mosquito from becoming a vector of this disease, like malaria or like dengue or other things. Now the tricky thing is that how can we spread that genes? How can we make all the mosquitoes in the wild or on the planet become resistant to those diseases? The way that people do it with CRISPR is that they use CRISPR as a tool for copying the DNA from one chromosome to other chromosomes. So with these mechanisms, you can propagate the genes in the populations at almost 100% efficiency. For a typical genetic inheritance, each of your genes has about 50% chance of getting to the next generation. But with this technology, it has close to 100% chance of going to the population. So this is one way that you can spread the genes that you create, or the gene that you select from nature, to the entire populations in the wild. This one is also a pretty controversial topic because this is a way that you can engineer the entire populations in the ecosystems, and it could have some consequence. We don't know, right? But the people who support technologies also believe that it's worth the risk because every year there are millions of people die from malaria or dengue or other mosquito-borne disease. So we should take that risk and and eliminate all these mosquitoes that spread the disease. There's also some research group that working on bringing extinct animal back to life. There's a group that called Revive and Restore. Basically, what they do is that they take genetic materials from fossils, from like the remainings of animals that they can find in the museums or other things, right? And they attempt to clone it into like modern days animals. But the problem with that is that sometimes the DNA is not perfect, right? So the way to get around this problem is that people use CRISPR to gradually modify the genetic materials of modern day animals back to the same version as the animals in the past. There was at least one or two research groups that working on editing the genomes of modern days Africans or Asian elephants back to the same copy as the mammoth and bringing the mammoth back to life. Just a few days ago, there was a big news in science magazines about bringing back Neanderthals. I'm not sure if you've seen the news a few days ago. So basically, they culture they make the mini versions of human brains, right? But before they make it, they edit the genomes, part of the genome of that cells, to become more similar to the brain of Neanderthals. Okay, and they use it to study, what is the difference between the human brains and Neanderthal brains. For the current technology, they just change at one or two locations. But imagine what can happen in the future once we can do it in a larger scale. And this will be my last slide. The reason why this is going to be a game changer is that the technologies become easier, cheaper, and faster. Right? In the past, when you talk about genome editing, there are probably a handful of labs in the world that's capable of doing it. It probably takes years. It probably takes tens of thousands or even millions of dollars to make genome editing. These days, the cost come down like thousands of times. The time it takes for editing some type of genome, it might be just a few days or a few weeks, right? And the cost, right now you can buy genome editing kits from many websites online for the cost of hundreds or thousands of dollars, right? So it's foreseeable in the near future that people can buy those kits and set up a lab and start do genome editing in their homes, right? So imagine what are all these possibilities and risks, So we'll end here, and I will open up for a question, or if I can open up for like one or two questions if you have. Thank you.
Is there any question regarding to this presentation by Dr. Park Poom? Please.
So you mentioned that you said it was going to like commercial use of genetic engineering for babies.
Mhm.
So are you inferring that it's going to be about 5 to 10 years before we actually can index the 3 billion polymorphisms in all the...
No. So okay. So there are two things, right? The question is whether or not we can edit the genome. Yes, I think we can already do it this day, right? The second question is that can we cure all the disease and can we make human perfect? I would say it would take like much longer times.
Okay.
Right? I think one of the key things that people misunderstood about CRISPR hype is that they believe that we're gonna cure all the disease very soon, right? Now, what I like to emphasize is that CRISPR is a tool that allows you to change a DNA sequence. It's like a pen and pencil, pen and eraser. Can you erase and change the sequence? DNA in your cell is like a book, right? You can go in and change the sequence. But another important component is that you need to know where to change it. You need to know what to change it to, right? Having a pen and pencil, it doesn't mean that you can write a poems or like write a textbook or like write a novel, right? Now, for that reasons, if you look at the CRISPR startup companies, most of them will focus on genetic disease that has a very precise and a very well-known locations in that is the cause of that disease. For example, sickle cells or cystic fibrosis or those thalassemias, we know exactly where on the genome we need to fix, right? But for many disease or for many traits, let's say like obesities or like Alzheimer or like how to make people smarter or taller, we don't know yet because there are so many locations on the genome that might be related, right? There are also environmental factor, right? So even if you have a perfect CRISPR technologies, you still can't solve that problem yet because we don't know where to change it yet. Yeah.
Is there any other questions? All right, and for this session, thank you very much, Dr. Phakphum Sapsunthorn. Thanks for sharing us about this. It's very inspiring. We'll let him take a deep breath for a few minutes as he's going to join us again on our next session as the moderator. Thank you very much indeed for joining us here, Dr. Phakphum.